say that can tell her you are sad.
Yeah.
Okay, so Jasna, please go ahead.
Okay, this is my last class, but it's also the most wobbly because I tried to make it related to everything I promised you. So I wanted to come through on my promise, but as a result, I had to do quite a bit of new things. So let's see how it goes. It's as new to me as it is to you. We started to talk about artificial life, and I feel like you guys have been spoiled over the last few days with like amazing talks on all kinds of topics, and at least all the stuff I've heard been really impressive. So I feel like you might be reaching saturation point. I don't know if that's true or not, but I thought maybe today we can just discuss the list that we had of the things you require to be alive. Leticia, you missed out on that, so we're going to do a bit of what is life like. Some things we already achieved, some things we still have to achieve. So this is my summary of where we are so far. The first two classes, what I tried to demonstrate to you is that there is something called the central dogma in molecular biology. Biology is based on the idea that the genetic code carries information to get you to protein structure, and that protein structure has a function. It's quite simple, and that's what this central dogma essentially proposes as one of the mechanisms for life. And I tried to show you that you can mimic that with colloidomers, which are polymers made of droplets, and they can fold into foldamers, which are those colloidomers that have unique solutions in the phase space of possible conformations. And what we try to do is to make those foldamers useful, for example, by making them, wrapping them into little capsids or making some function for them that's quite simple. Even once you're done with the building blocks like protein-like things, you want to self-assemble them into compartments. You want to build the hierarchy. So the next level is about what can you do with supramolecular interactions? How can you mimic those? And I tried to show you this idea that you can circumvent the curse of dimensionality, as Mathieu called it, by going from 1D to 2D to 3D. So you're funneling the landscape in this way. And we tried to, at the end of the day, add dynamics to it. And we added dynamics by putting some enzymes, so borrowing from biology, to make logic gates between those droplets. Who likes who and how much in space and in time? Can we switch interactions on and off? Can we make them spatially segregate? So we've done a lot. I'm going to say that that's metabolism because the food for the droplets is the sticky strands of the DNA that are made by the polymerase, and the on and off switches and the segregation of the color blue-yellow or things like this is going to be their useful consumption of food to energy. So it's somehow, for those of you who are familiar with biology, this could be a mimic of the differential adhesion hypothesis or something like that. How do cells segregate into different tissues, for example? Okay, at the very basic level. What I didn't do at all is I didn't show you anything about self-replication, and I didn't show you anything about the ability to evolve a structure, and I didn't show you anything about motility. My favorite moment of this conference so far is when one of you said that a signature of life is the fear of death. And I was thinking, there's just no way we can demonstrate that, except, isn't that always present? So, again, the goal of Rachel. So that's where we are right now. So let's begin with what we haven't done yet. That was just a little recap. And this is a paper I really like. It's done by Penrose, and most of you, or some of you might know about Roger Penrose, famous physicist. But actually, this is his father. So this is a 1958 So, 70 years ago, people started to think about, can you make a mechanical model of self-replication? What's the simplest way you could possibly do it? And I love this paper. It says, perhaps the most remarkable feature of living matter, as opposed to inanimate nature, is the power of self-reproduction. And then here, what's funny is it says, it happened that my colleague, Roger Penrose, it's not really his colleague, but mentioned that, blah, blah, blah. So I want to make one point that is very fascinating to me, at least. If you take a really simple example of these building blocks, they're just a bunch of shapes. You put them in a line. This is a real experiment. I wish I had a demo. And you jiggle them about, nothing will happen. But if you tilt the thing, or if you shake it vertically, what you get is that they stack up against each other, and you seed a crystal. And you get growth, and you can grow a line of a perfect crystal. And then crystals do not qualify as self-replicating because they grow essentially algebraically with time. There is no exponential growth. So the question becomes, what can I do with a crystal to make it grow exponentially? And what you can do is really quite simple, and it's mentioned in this paper. Another reason I like it is you can take, this is a better mimic, a hammer and smash it and keep growing those crystals, and that will give you exponential growth. So the simplest example of self-replicating, exponentially growing system is to make a crystal, smash it, grow it, smash it, grow it, and so on. Instead, what you can do is you can make the building block a little bit more funky. And the way in which it's funky, you need to have good eyesight here, or you can just trust me. If you take the same bunch of building blocks, shake it horizontally, no one likes anyone. Then you do another round where you tilt it, and you see that there's a special groove that B can bind to A. And now you've made this dimer, and if you keep shaking, keep shaking, keep shaking, they all stack up, but it's no longer a crystal, because if you've got very good eyesight, you see that only B likes A in a sense of bonding, but everybody else, there's a mismatch. And so when you stop shaking and you look at how many things you've made, you find that only the ones where B is next to A have actually made the first replica of the seed that you've got there. And then you can play around with geometry to make this groovy stuff in different places. And for example, this is the exact same protocol, but for an A-B block. And you can do the same thing. There's a mismatch. And again, you can make a duplicate of the A-B block. So it's sort of the first publication showing a mechanical principle for what you might need for self-replication. And what's the minimal ingredients is you need some irreversible bonding and some reversible bonding. And I'll make that more clear as we go with real examples. So where do we know that exponential growth exists? If you want to borrow from biology and billions of years of evolutions, you say, okay, I know how to do a polymerase chain reaction. I know how to take already made double helix of DNA with its Watson and Crick pairs. I know how to heat it up to death to make them fall apart. I know how to borrow from the machinery of biology by having an enzyme called a polymerase that can essentially write the rest of this single strand, the matching pair, by taking bases from the solution. That happens only after I cool the system down. And then what happens is I recruit all these bases from the solution, and what do I end up with? I start with one strand, and I have 2 copies at the end. And I do this 30 to 40 times, and the PCR machine amplifies any DNA. This is, for example, the basis of the spit test, whether you have the flu or COVID or whatever. It's like you have a little bit of DNA, but then you do this to it, you amplify it, now there's lots of it, and so you can do manipulations with that. So, is this self-replication? Yes and no. It is, because there's exponential growth and you're making copies. Is it cheating? Yes, it's cheating, because it's not really taking inanimate objects and making them artificial life like. So we would like to do something that's more, less borrowing from biology. Okay, so just for fun, I thought it's interesting to look at the atomic structure of these polymerase. That, imagine that this is a soup of bases, and then this polymerase green guy is taking in and making DNA out of by recruiting the bases and growing the complementary pair. That's what the machinery looks like. Okay, so now we come to the fundamental statement, which is, if you're in equilibrium, you're dead. We already started the course with that statement. And Louis Pasteur, he says, omne vivum ex vivo. So it says, life only comes from life, and you can't really do anything unless you rely on other things that are alive. It turns out that's actually a true statement. Viruses that can self-replicate need a host, but it turns out we also need a host. We also need to eat plants that give us things, molecules that we couldn't otherwise make ourselves. So there is no example, at least Paul Chaikin, who is the, author of most of these inanimate self-replication papers, and my colleague next door, he's telling me that he asked many biologists and no one has been able to give him an example of an organism or even a piece of it that can sustain its own life without the presence of others. Now, because you're smart, you're going to say that's got to be wrong in time, because when life began, there weren't any others. So how did it begin? So clearly there's a different machinery. This is about what's around today. But clearly there was something where life was sustained without the presence of others. Right? I don't know. Maybe. So for self-assembly, we go back to droplets, we go back to colloids, we go back to the idea that these blue and yellow particles that you were folding, now they're red and green, and they have DNA in between them. And now we're going to use the sticky end red and sticky end green that come together to hybridize the droplets together. They're going to be the element for self-replication. And I'm going to explain how. So just refreshing your memory, this black tether here does nothing. That's just the arm. And then when two droplets meet and greet, they bind through the DNA, and that's the hybridization of these two strands gives the free energy for adhesion between the droplets. Okay, so that's just, you knew that already because that's revision. And now this is the basic replication scheme. How do you self-replicate? You need a seed. So we create our colloidomer chain the way that we did together, actually. And in this case, you make a backbone here, A, B, A, B, B, A. That's the sequence. So this is the mother sequence. Now we need a daughter sequence. And one thing you'll notice is that you want something, let's do it, that is complementary. So A will get to A prime, B will get the B prime strand, etc., etc., etc. So you could have a soup of droplets, and they would only bind to their correct pair. Now, you have horizontal bonds, and you have vertical bonds. But what's going to happen if I now heat this thing up above the melting temperature of the DNA? What's going to happen to my, even if the backbone is stronger and made of other DNA, what's going to happen to these vertical and these lateral bonds if they're alike? They're all going to disassociate, and we're going to go back to square one again. So we need an extra ingredient, and this is key for self-replication. That was UV light shining at this thing. You rise the temperature, and now we want that there's an asymmetry, that the vertical bonds fall apart, but the horizontal bonds stay intact. And so how do we do that? Well, no. How do we do that? We have two types of DNA bonds. We have, remember, this is again revision, palindrome sequences that read forward and backward the same way. And we have Watson-Crick pairs for the vertical guys, right? And the beauty of this guy here is that there is a little zone in here which has a special molecule, organic chemistry molecule, I'll show it in a second, made by another colleague at NYU, Marcus Weck, that can essentially switch on the bond when you switch the light on. And so you can make irreversible covalent bonds by cycling light, okay? We still need the cycle of temperature too. So here is, you guys are not chemists and you don't care, but I'm still going to tell you about it because you should be intellectually enriched. So here is the idea. You shine light. What happens to these delocalized rings? Essentially, you make this square here. And if you look at, this is the zoom in of the base pairing, adenine, thymine, guanine, cytosine. So that's your regular alpha helix with the right bonding. And this is the XX on the other slide, which is this fake organic molecule. And when you put them together, it essentially changes to make this little square here. And that's what gives you this irreversible binding energy. Okay, so I promised you that I would show you how you can grow these in Washington Square Park. So this is from NYU. This I thought was a cool fact. It just so happens that to make the chemistry work, you need about 30 degrees Celsius in difference in temperature between day and night. And that just isn't the case in New York City most of the time. It does happen. But you can trick the system by doing the experiment in dirt. And when I say I or we or anything like that, what I mean is Paul Chaikin's group together with Ned Siemens' group at NYU, and I'm reporting on their results. So why do you think that it helps to have dirt next to you in order to achieve a larger temperature difference? You're not doing anything with dirt. You're just putting your experiment on dirt. Yes, it has the capacity to retain heat. So what you end up with is that just like when you wear a black T-shirt, it absorbs heat, and so you end up with hotter things, and so you end up with larger gradient in temperature. And then there's sun, of course, that comes in and out. So you want, the idea is that you start with inanimate objects, your poor man's DNA strands with colloids. You give it some daylight and some nighttime. You give it some fluctuation in temperature. And you come back, and what do you want to find? You want to find exponential growth of the material with which you started. That's kind of crazy as an idea that that would be all you need, right? And we weren't able to do it, actually.
Can I ask you a question, which I think is very naive?
You can. We're trying to do this still, so I'm giving you the chemin, but actually it's very complex, so we were not able to do it yet.
Okay, no, but my question is very, I think, very naive. Why do you need exponential growth? To sustain a population, or because you could think about just keep it constant, but then you would need death. Is that what you mean about exponential growth?
I mean, what you'd like for exponential growth is that if you double in every, that I start with one monomer, and then maybe I come back 6 cycles later, and I'm so smart about the way I crash my crystal or my strand that I can get a 10-fold increase, and then I have 1 million after a few cycles. So now I have a real way of, the true reason is I can make different populations compete against each other, and do Darwin. I mean, I haven't told the evolution goal, but I really want to compare who survives, the survival of the fittest. I mean, that's afterwards. First, we have to grow one species. Any other questions? Yeah.
Do the things in the soup that you have combine with each other without copying your polymer as well? So apart from
do you
copying
get non-specific interactions? Depends how good of an experimentalist you are. So you design the system in such a way that if you did it in a computer, it would be perfect.
You don't have a polymerase, a sort of machinery that brings the thing and creates the reaction.
That's an excellent point. We don't use polymerase. We're not borrowing from biology. We're simply saying, can we design the DNA sequences to start with in such a way that we would make this happen autonomously? So that's the way we're kind of arguing why we're even doing this. Otherwise, we have PCR already, right? Okay. That's how Paul argues it. But we are trying together to do this with particles, and it's harder. But let me show you a thing that does work. This is already remarkable. So like I say, in Paul and Ned's group, what they did was just imagine that these were droplets. All of everything I've said so far still holds. What you can do is you start with some daughter tile, so some monomer. And what you do is, again, you have the same idea. When you shine UV light, you only stabilize, let's say in this case, the horizontal bonds. And when you heat it up, the ones that come apart are still weak, and those are the vertical bonds. So what did you do? You started with this guy, this dimer, blue, and after a cycle of this ladder growth, that's what we call it, is ladder growth, you end up with 1, 2, 3, 4, 5, 6, 7 daughter tiles. So that's an amazing reproduction method, because it even beats the exponential growth, if you like. You're not going 1 to 1. You're actually growing more than the one you wanted. Okay, so let me show you. This is a real experiment with real data that really worked. So for example, this is the cycle number of the days and nights, temperatures going up and down. And this is the measurement of the amplification factor. And so don't worry about the different curves. There are different amounts of monomer from which you start, and there's all sorts of detail that A, I wouldn't know, and B, I don't have time. But what's fun about this number is, you see, I like 1 to the 6. If you have 10 to the 6, you have 1,000,000 up here, only after 6 cycles. So you start with 1, you end up with 1,000,000 after a week or so. So that's pretty nice. They really made it work right out there using just the natural 24-hour clock. Okay. So that's my first demonstration of the self-replicating equilibrium system with an inert set of chemicals, no enzymes, no biology, and achievement of the exponential growth. I'm not going to go in any detail on this, but just because we got obsessed with folding, some of us, you can also do a more fancy version of this if you start to use these crosses to assemble in 3D by a lot more DNA interactions. You can even start to make boxes, and you can make ways in which they can then self-replicate. So this is the beginning of a world within which, you know, inanimate structures could be grown. I mean, at least the proof of principle is there. Okay. Now comes the fun part, what you asked. Like, let's see how we can do selection. Is there a way to make 2 tiles compete and see which one grows faster? Is there a way to observe Darwinian survival of the fittest in such a simple system? And here is a way that they designed, which is to say that these 2 crisscross tiles have 1, 2, 3, 4 sticky ends that can come together. So this is when they're unbound, this is when they're bound. You see it here. And what you might notice, just look at these letters, T, T, G, G, C, T, finishing with GA. You have the same strand here. The red tile and the blue tile, they have the same sticky strand. The only difference is that this blue guy, one base, is different for every one of the 4 sticky ends. So it's a tiny amount of difference. You might think it shouldn't make any difference on the reproduction rate because it's really just one letter in the sequence that you're changing. But that's not going to be the case. You will see that. So this guy is more favorable. This guy is less favorable for the binding. We repeat the experiment. Can we see that one of them grows faster or not? And here comes another, let's say, bombastic result, because it sort of shows you that you can think of this as 2 species competing for food. What is the food? The food is the connecting sticky ends. So whichever one gets more of the connecting sticky ends gets to reproduce faster in this soup, okay? And here's the result. The one that is preferred, the one that you saw on the left, if there is enough food for everybody, this is the number of connection strands or the concentration of connection strands that you have in your soup, then there's no speciation. There's no survival of the fittest. Everyone gets their food, and you roughly make the same. There's 1.1 is the ratio of the 2 different tiles that you end up with. But if you starve them, and this is really fun because it really shows you can wipe out a population very easily as long as you starve it for a couple of generations. And what's remarkable is to demonstrate that on a crisscross DNA origami. And so here it is. And I want you to focus on the fact that the growth rate doesn't look spectacular, but if you look at with the limited supply of connection strands, the ratio of the 2 strands is almost 20. And so you can really make them die out by very tiny preferences and how they stick together. So it's kind of a remarkable result to show that with such a simple system. Any thoughts, questions, concerns? No? Okay.
Yeah, I have concerns. And let me
Yes.
ask you.
That's
So in the
good.
first part,
Yes.
should I retain that to replicate a minimum ingredient is irreversibility? Can I say this?
No, you need 2 distinct bonds. One that is irreversible
Yeah.
and one that is reversible.
Okay, that's
Such that when you heat it up, they break apart, but the ones that make the daughter don't break apart.
Yeah, and then
you don't want to go back to the soup.
Okay, and the second, in this part, you need to have one of the species which is more adapted somehow.
It's a better fit, meaning that the free energy of binding per bond, per Velcro, is essentially better in one of the cases than the other. But it's a tiny amount, so you might have thought it's not going to matter that much, but actually it's enough because you're growing over these several generations.
I have a quick naive question.
Yes.
Is that ratio a ratio, the A1, A2 over B1, B2, isn't it time dependent or something, or does it
this
saturate?
is after the whole thing is done.
All right, so it saturates.
Yeah, we don't do they haven't done it more than 10 cycles, I think, ever.
Okay.
So yeah, it's at the end of the experiment. You're right, it's very time dependent. So, survival of the fittest, that's kind of nice. In Darwinian evolution, species that are more suited to the environment are more likely to survive and reproduce. And there's your tall-necked giraffe to prove it. Okay, let's talk about something else now. And they have a different, what's it called? A different format, so I didn't want to reformat everything. I thought you could be patient enough. So that's kind of done with whole of the lifelike material based on self-assembly, and this is completely different in a sense that we're going to use the same droplets, but this time we're going to make them swim. And since you guys had a lot of lectures about active matter and so on, you can add to your palette of things that can move around by consuming energy. And we're going to talk about how you can make things swim. So going back to biology, if you look at paramecium or bacteria or sperm, or you look at what are the minimal ingredients to make something swim, you need a source of energy and you need to dissipate it into viscous drag. That's the main thing you need, and you need some break of isotropy of the interaction with the environment. I think Anton had a beautiful video that I might ask to steal for future talks of the clam, or the idea that you need to have an asymmetry in the flow such that you can actually get to eat. I thought that was a pretty cool demonstration. Okay, so then inspired by these systems, real biology systems, lots of soft matter physicists said, can we build something similar? And you've seen other examples too, but here I wanted to highlight you can make nanorods that can really swim mimicking sperm. You have Janus particles where you have peroxide interaction at the interface that makes it polarized, so that's enough to give it a direction. And then this is the system we want to talk about, which is the idea that you start with an oil droplet in water, and it starts to swim. And that's a really weird thing. And I want to spend a little time explaining why. Before I do that, though, I wanted to highlight a second important phenomenon. So one is how do they swim, and what makes this energy into viscous dissipation special about droplets. But the second one is, this is probably the most famous movie in which you see the neutrophile chasing a bacterium, and finally it actually gets to engulf it. And what's that based on? That's based on signaling. And that's based on the fact that this guy is able to do what we call solute-mediated sensing. So there are concentration gradients that the neutrophile can sense and say, I'm going to go this way instead of that way. And in this case, it's called a chemoattractant, but there are also chemorepellents, et cetera, et cetera. So one other thing I want to talk about for the rest of my talk is how can you mimic anything like this again with a very simple system? And so I'm going to be rude again and say that the living cell is like an inanimate droplet. And I'm going to try to argue that we can measure these solute-mediated interactions by using laser tweezers and such like to try to infer what kind of memory do these systems have of their past, and how the memory of the past influences their decision making. Okay, so I wanted to tell you a story, a 1-minute story, which is that everything I told you so far is kind of like I didn't like it because the idea was we start with theory, we predict what kind of sequences can fold into what kind of structures, and then we say, oh, the theory says it should work, protein folding problem in this instance is solved, so go and make it in the lab, and then we go to the lab and then it works and then we're happy. But theory that experimentalists test is not as exciting as when you go to the lab and you see a phenomenon and you're like, what the heck? I really want to understand what's happening. So most of our work is actually done this way, even if we tell the story as if it was done the other way. But this is a nice example because a postdoc of mine, Martin Haase, he came and said, yes, you got to come to the lab The only thing I did was to put some oil in water, and I added some soap. And I looked down under the microscope, and the droplets are swimming. And I was like, no, that's not true. That cannot be. And we had these long arguments, and I kept saying, you haven't washed something properly, or, I don't know what was. So this was the video he showed me, and he was like, this looks weird. This droplet is really moving in a motility type that doesn't really even look Brownian by eye. It's sort of curling, swirling, doing things. And I really think I discovered swimmers. By the time we came to a point where, like, this is fascinating. We should understand this. We should study this. There was a paper by Olivier Dauchau's lab, who is a friend of ours and a friend of mine and colleague and everything. And it was exactly about that. But we still got the kind of eureka moment in the lab, because we still discovered it for ourselves. So it's not that we hold the discovery of this, Olivier does, but what was nice is this idea, let's study something that we really don't understand. So let me introduce you to this system a little bit more. If you take, it doesn't matter that it's a nematic liquid crystal, it's still an oil droplet in water. If you change the temperature and you look at the XY trajectory, what you see is that there are regions where the droplet goes straight and then it starts to do this curling. If you change the viscosity of the solvent, what you find again is you go from ballistic to some kind of chaotic motion. So lots of people chained on to start to discover that these droplets really swim in strange ways. You vary the size, you see random motion and helical motion and straight motion, and so on and so on. And we decided to study this in a system that's similar in spirit, but we wanted to see what happens in a lifetime of a droplet. If we inject a big droplet, we make it swim. Is it going to end in death? What does the death of a droplet look like? And so on. So I'm going to end with that. I'm not going to tell you yet. So what is the system? The system is really simple. Diethyl phthalate, which is DEP, is just an oil. It's a slightly polar oil. SDS is the most common surfactant there is. Water is water. And so it's a three-component system: oil, water, and soap. Nothing else. And you make the droplet 200 microns, and I'm going to play the movie for you again, just so you get convinced. These droplets really swim, and they swim very fast. I think they swim something on the order of 20, 30 of their body sizes per second or something. So they really whiz around. And how fast they swim depends on a lot of things, but it depends on what is the fuel here, what is the engine. So in all the other examples, including the movement of flagella, the bacteria, they have cilia, they have mechanisms to make themselves propel. In the case of active matter like Janus particles or colloidal rods, you always have interactions at the interface that you engineer to give a little rocket ship to propel forward and to make the asymmetry of the particle. Here, there's no anisotropy. It's just a fully isotropic sphere. So why does it swim? And so we noticed first that if you work below the CMC, which is the critical micellar concentration, which is the concentration at which the surfactant itself starts to aggregate into these little clumps, then the droplets dissolve into the solution. So you start with a drop, it shrinks, it shrinks, it shrinks, it disappears, but it doesn't swim. But if you work above the CMC, they start to swim. So somehow, and if you work below this concentration, then nothing happens. They just sit there like a regular emulsion. So it's, yeah?
Understand that you have water,
You have water, and you vary the amount of surfactant.
which is just soap.
just soap.
And then you
Put a
put
little-
some droplets of oil.
And you
Is that
put
you do?
Yeah,
That's
you could do this experiment.
Okay, I
Really,
see.
you can do it at home
okay. I see.
and convince yourself that it's true.
And do you control the size of the droplets?
By
You
using
then-
a micro pipette.
Okay, that you do by hand. Okay.
Yeah, by hand, yeah. Okay, so that's the phenomenon, that's the fun stuff. And then we said, we think we know what's happening, and I'm going to show you first what we thought was going on and how we then tested it, because, I don't know, I tried the story the other way and ran out of time. So it's like, I want to tell you what it is, what is the engine, and then how to test the hypothesis. So we said, what could be happening is that consider this oil droplet isotropic at time t0. It starts to shrink. And as it's shrinking, why is it shrinking? It's because the reaction at the interface is that these molecules of oil, the yellow, is getting eaten by the micelles. Because the micelles are the surfactant molecules that have the hydrocarbon chains pointing inwards. And so they like to be oily. And so they swell the micelle in size. And so the free energy thermodynamic equilibrium would be that everybody is in micelles, and there is no more droplet. And so that's what's driving the dissolution process, is that this oil is slightly soluble in soapy water. And what's interesting about, if you think about equilibrium around the droplet, is you have some fluctuations, right? This interaction of reaction with the micelles, you have collisions, it's being bombarded. At some point, there is an instability, and you end up with an orientation of the surfactants at the interface. But how do they orient? They orient because of one key ingredient. The critical micellar concentration in pure water is, so here is 9 millimolar, whereas in dirty water, it's much lower because they like it better when they're filled with oil, right? So less monomers go into here than they go into there. And so the equilibrium between the interface and these empty micelles has a higher concentration of surfactant than at the back of the droplet. What does that mean? If I give you an interface and I give you less surfactant to cover it with, what happens to your interfacial tension? It goes up. If I give you more surfactant to cover the interface, it goes down. So now you have a difference in pressure, interfacial tension. You can think about it as a pressure at the back and the front. And so you have a delta P driven by the fact that these guys are more dense here than they are there. Now, what's going to happen next? If I have a sphere, and here it's crowded, and here it's not, well, if they want to equilibrate, these guys from here are going to go to the back. So you're going to get flow. So the flow of these surfactants from the front to the back of the drop, and by the way, the choice of what is front and back is random, because it's just wherever the collision took place. You end up with having flow of the surfactant towards the back. Because of that flow, you also end up with these other flows. And so now the droplet is moving towards what? Pure water. Because that's where the pressure is lower, right? So there is an interfacial tension-driven engine, the speed of which should depend on, for example, the change in the CMC between the front and the back. So all of these things we can test. We can test how does this depend on how many micelles we put? How does this depend on the CMC? How does this, so we can actually test these ideas in the experiment. And that's what we ended up doing. So let me ask, is that clear? One of the things you might think is that you just equilibrate very fast. So why don't you? It's because the time it takes for you to relax is slower than the fast motion of the drop, which is again in pure water. I don't know if you've ever played this game, Snake or something, but this is a little bit like this phenomenon will become more and more like a snake.
One thing that bothers me in this idea is the following. When I want to take out the oil from, I want to wash my dish,
Yeah.
and then
This
I put
works.
soap,
Yeah.
right, to, let's say, to dissolve oil somehow.
Yeah.
But now you are telling me if I put too much oil, I will not do that. I will not dissolve if I put a lot of soap, sorry,
If you put a lot of soap
I
and
will
you
not
have,
dissolve my oil anymore.
it will. The
Ah.
free energy minimum will be to make your soap dirty with oil. So you'll have swollen micelles of your soap with little bits of oil in it, instead of your big blobs of oil on your plates.
Okay, but then it would,
I don't know. Something like that.
okay.
Wait, is that confusing or not?
A little bit fun, but I mean, go ahead.
Okay,
Then
but this is the Ferrari of swimming droplets, because in fact, the dissolution of this specific oil in the water is way faster than any of the other experiments that I showed you before. So let me show you how fast that is. Yeah. Yeah, sure. This is not trivial at all, and it's, perhaps even still up for debate. Not for me, but maybe for others.
How does the collision time scale kind of enter all of this? I guess if the micelles collided with the droplets very often, I guess you would recover a fairly,
So that's super
isotropic
interesting. So as you increase the concentration of micelles, which increases the collision rate, you could argue it both ways. You could think that, you have like central limit theorem type of idea, whereby this reorientation of the micelles at the interface becomes less important. Or you could argue that, in fact, the collisions are more frequent, but it's enough to polarize the droplet, and so it becomes more chaotic. And so I don't want to tell you because I want to show you.
Okay. Sounds good. Thank you.
But you can think of it both ways. Yeah? No? Okay. Good. So here's a couple of things you can measure. Don't imagine, this is what we did. We inject the droplet here at T0. The contrast is not so great. So again, if you've got good eyesight, you might be able to see blurry things around the yellow. Do you see like a blurry gray area? That area is large here and small, progressively less wide, because this is the fluorescent dye of the dissolving oil. So this photo was taken at time t 90 seconds. So this is the diffusion of the oil away from the droplet 90 seconds later, okay, at T0 here. At the time that the photo is taken, there is no diffusion because that's the moment you took the photo, right? So what we can do is kind of cool. We can model this as a Gaussian, basically diffusion front. And from the fit to the data, we can estimate what the diffusion constant of these oily micelles away from the droplet is from the fluorescence. And we get a number, 72 microns squared per second. And then you can ask, what's the size of a particle of that diffusion constant? You get 3.4 nanometers. And lo and behold, that's known to be the size of this micelle. So that was a sweet moment in experimental physics where things make sense. There are other moments when things don't make sense. And the other thing and this becomes very important for the input parameters of the theory. Then another thing you can measure is how fast the droplet shrinks. This is the radius, and then this is over time. And you see that in this case, you have 0.3 microns per second, the shrinkage rate. And so that's another important parameter in the theory because it's going to be one of the parameters. Okay, we're in the low Reynolds number. I'm not going to go into it. I'm going to skip a few slides. That Leticia, I can just say things now. So you can think of, can we model this as an active Brownian particle? If we could, then it would have a velocity for the ballistic part of the trajectories, and it would have some diffusion of the angle, which is based on God knows what. Why God knows what? Because temperature is completely irrelevant. in this system. So if there was a temperature that was mimicked here, we would have no idea where it comes from. And so that was the fun part. Why does temperature not matter? Because a blob of 100 microns in diameter doesn't care about temperature. It's just not. It's a macroscopic object that's moving around by magic. Okay. So if it was an active Brownian particle, the test could be, is it ballistic at short times and diffusive at long times? We tried that, and in fact, for a little while, it worked. What do I mean by little while? If the size of the droplet remains roughly constant, and I do the time very, very short, like 1 second out of my 200, then it doesn't work so badly. I vary the concentration of the surfactant, that's what you wanted, and I measure this as slope of 2, so it's essentially only ballistic here, and then you start to observe some shift to slope 1, which is the diffusive part of the curve, and you see that it becomes diffusive at progressively earlier times. So Anton, this is your question. One interpretation at very, very short times, the collisions matter, so the more surfactant you have, the earlier you become diffusive. Okay, so that's one. I'm gonna say something later that will go the opposite way, but at short times, this is true. Make sense to people? Okay.
A quick question about
Yeah,
your
sure.
equations just 1 or 2 slides ago. Doesn't translational diffusion, isn't it important? I noticed that you had that
Yeah, there's
white noise.
rotational diffusion and there's translational diffusion. It depends on what the diffusion constant of these things is, and since it's not thermal, you know, the question really becomes on what time scale does it become relevant. So for example, for colloids, you never even see it because you need to have femtosecond cameras or something like this. Here you will see that actually, if we look on longer time scales, we can measure the diffusion. So you see a diffusive, a ballistic, and then a diffusive regime. But the story is going to change as we look at longer times. Okay. So again, summarizing what Anton asked, which is that tuning the noise or the persistence time or the frequency with which the droplet changes orientation essentially goes to slower and slower times. There's some kind of a power law that we have no idea what it means. And it's, of course, over less than a decade, so who cares, right? But the idea is just that you have a 4 orders of magnitude decrease in this time, which we interpreted to mean that the collisions matter. This is the more fun result. If you want to change the critical micellar concentration without changing anything else in your solution, so when do the soap molecules clump together into micelles, you change the salt. It's a kind of trick that exists in chemistry. And look what happens. You go from, you know, very little salt to a lot of salt, and what you see is that the Ferrari, the 500 micron per second droplet, almost goes to 0. And if, in fact, when the delta CMC, so in the model, you would always end up with a finite thingy, but in the data, there is a delta CMC below which you don't even swim. So you can switch off the engine by adding salt. That's kind of fun. So all of these things are a bit consistent with the theory that we proposed. Okay, so that's just for the engine, how we think the droplet moves. Now comes the fun part, for the snake part. If you're really good at the game, you end up avoiding your own tail very, very well, and you get really high scores, and you end up in the Hall of Fame. That's what these droplets seem to do. They end up having a non-Markovian random walk in a sense that on some time scales, they do cross their own path, and on other time scales, they do a self-avoiding random walk perfectly. So we decided, let's study that really in detail, and let's think about how we can implement memory effects into the analysis of these mean square displacements of these trajectories. Yeah.
This non-Markovian random walk, maybe I'm totally missing something. I know that there's been some work on, like, if you have like a Brownian like motion random walk with something that has some volume or surface area, like whether or not it's happening in 2D or 3D has a very strong effect on whether or not it will probably self-intersect. I mean, I guess
It's a big difference of the scalings in 2D and 3D, for sure.
right. I mean, are you always looking at, I guess, 3D, or is it ever sort of on a
Everything
sheet?
is in 2D
Okay,
in the
2D, okay.
experiment because this world is heavy. So
Okay,
we really only look at the droplet in 2D.
okay.
It's actually a complicated story in 3D. I'm just going to leave it at that for now. So let me show you the data. This is you pick one STS concentration. We show you that we do the experiment many times. Now we're doing it for much longer than 1 second. Before, I was showing you things that are really limited to a time window that's almost invisible on this scale. It's so small. And we see self-avoidance, for example, here. You see it very well. We see crossing much more so this is the beginning of the trajectory when you're big, and you shrink, you shrink, you shrink, and this is when you're small, and then you start to cross your own tail. And first thing you observe, of course, is that the way this trajectory evolves in time implies that if you look at the MSD, the ensemble average and the time average are not the same. So you need to be very careful how you analyze your data because, in fact, depending on which radius or which time you pick to analyze the mean square displacement, you get a different result, okay? And so that's the first signature of non-Markovianity in your data, is to say, is the MSD the same or not? If it's not, you have a harder job to do the theory. Okay. Second stage is how do we actually take this into account? So let me explain for a minute what the assumptions of the model are. So we say that the droplet is essentially, you can think of it as, the analogy would be, imagine that the droplet was full of sand, and then as it moves, it's dropping sand, and then it makes a little pyramid, right, a little groove. And then that's like a landscape that it's carving for itself, and that's evolving with the diffusion constant that I described to you. And as the landscape is evolving, I'm moving on it. So the big point is that my motion is coupled to the concentration gradient that I'm exuding. So how do I take that into account? Well, I can think of this, I can describe the concentration field of these swollen micelles over time by the diffusion equation. And I can think about the oil expulsion from a droplet as having a source. And that source can be a delta function if I'm lazy, or it can be a Gaussian with a finite size of the droplet to take into account that you're really leaking here and not from the center. And that's actually important for the experiment. And then I have another equation, which is the equation of motion. Let me just put which is the equation of motion for the droplet. So I've got my drag. I've got coupling strength between what? Between the gradient of the field of the oil and the position of my particle over time. And so what you see is that you can make this a non-Markovian model by essentially integrating over all time the whole history of the droplet trajectory. And of course, I have some noise term that I told you I don't know the source, but I'm just going to introduce it there for now, assuming white noise. And so I have some noise amplitude, I have a coupling strength, and I have this essentially a response of the force. So you can think of this as, what are the forces acting on my droplet? There is the drag, there is my fuel, pushing me forward, and I have a response to the gradient that I've created. And I need those three ingredients to be integrated. And how do I do that? I do that by integrating over all the path that the droplet has taken and multiplying here by the gradient of the concentration field. Okay? That's the model. All good? Okay. Now, the droplet size is time dependent because of shrinkage, but unfortunately this term, the droplet size plays a role in all the terms in the equation. So the math gets a little bit messy, but you can still have closed solutions. So I just want to show you for a moment. You can take into account how the, for example, shrinkage rate depends on the size of the droplet, or how your Gaussian kernel source term depends on the size of the droplet, or how the viscous drag depends on the size of the droplet. So you can do this mathematically for all of them, and you can basically say, if I take into account the influence of size on all this term in my model, do I reproduce the same trajectories as what I got experimentally? Okay.
Jasna, can I ask
yeah,
you something
sure.
quickly?
Sure, sure.
Yeah, so that's very interesting. You have this particle moving and leaving a trail, and up to some short, I don't know, time scale, it avoids crossing its trail.
Correct.
Do you have any way of estimating this time scale in which it avoids it? Is that a characteristic?
Yes,
a thing with
absolutely.
characteristic time that
Everything is very physical at the end. There is a force with which you are essentially, you're balancing what the gradient of the concentration field of the oil is. So once that becomes shallow enough in my model, well then my droplet has enough oomph to cross it without needing to do a U-turn. If it's a very sharp wall, well then the droplet comes in, feels the wall, and says, oh, I'd rather go the other way.
all right. And you have a way of controlling this experimentally, for instance? Is that just a tuning parameter for you?
So what we do is we build this model. We come up with fitting parameters to the model to estimate, for example, the coupling strength between the droplet motion and the gradient force, or the gradient of the concentration. And then we ask the question, how do these parameters depend on STS, viscosity, salt, all these different things? So we get intuition for
Got it.
what affects
Okay, thank you.
the droplet motion. I'll show you. I mean, this is where I'm going. Anyone else? No? Okay. All right. So this is a little bit iffy. Maybe people are not going to like it, but let's see. We make an assumption that the response to the concentration gradient should have an R-cubed dependence, or a volume dependence. And I'll explain a posteriori why we think that was a fine thing to do. For now, just bear with me. And, sorry. So that was here. So we make an analogy with electrostatics, and we say, we know that the force is determined by the electric potential gradient and the amount of charge. So here we argue it should be the same. It should be the concentration gradient and the amount, the amount of oily micelles that we start with, okay? And so it should scale with the volume, if there was an analogy. But then you could say, what gives you the right to think about solute-mediated interactions in terms of electrostatic analogies? And there I have a nice interlude, which is that we did a different experiment on the same system with laser tweezers, where we bring two droplets together, like two, you know, they're exuding their concentration gradients, and we force them together, and we let go, and we measure the speed at which they move apart. So we measure this repulsion speed in these experiments. And what we find is that the slope here does scale like the inverse square law. So we say, okay, there is some consistency with this analogy of between charged particles, so maybe we can take that further and say it's okay to think about it as a volume term. Okay. So the size of the droplet, the diffusion constant, the shrinkage rate are all measured from experiments. So the blue are the good guys. These are the bad guys. We don't know what the noise is, so we need to fit that one. And we don't know what beta is, which is this strength of the coupling between the field and the particle. And so, so what we do is we take the mean square displacement of the ensemble average data, not the time average data, because that's something we can compare between experiments easily. These are the input parameters independently measured. And then we can fit the data with the model to obtain a value for beta and a value for sigma, which is the noise, the effective temperature, and the coupling. And if we use the fit parameters to then generate trajectories, I hope you can all agree with me that the agreement looks really nice. I mean, we get this time evolution from the theory, where the crossing probability increases with time, and where they become progressively less ballistic the smaller they get, right? And we used AI or things to say which model does better, and this, and the other. But at the end, I think you can just see it by eye. So what did we learn?
There are just 6 different initial conditions in this.
We show 6, we do 1,000.
Okay, there is no difference between them, just initial condition.
Just to show you, we have lots of data that all look alike in some sense, but there is randomness in all. So what did we learn? We learned a bunch of things. We learned that, for example, we can look at the drift velocity of these droplets, and it seems to decrease with time. That's a bit counterintuitive. You might have thought that the smaller the droplet gets, the faster it goes, because the drag will be less. But in fact, we see the opposite. So why do larger droplets drift faster than small droplets? It's because the engine of the Marangoni flows is way stronger for a larger droplet than it is for a small one. So in fact, that's a first little lesson that you get, and there's a nice scaling that you can look at to look at energy consumption. You also learn something else, which is that if you look at the noise term as a function of time, the smaller you get as you shrink, the larger the noise. Well, that makes sense. If you're smaller, whatever the thing that's perturbing this flow is affecting you more. So the smaller droplets feel larger noise, larger amount of fluctuation, even if the temperature is the same. Do you see what I'm saying? For the same temperature, a smaller droplet will have larger fluctuations. That's it. Then we say, what's the effect on the turning probability if we change the surfactant? Well, guess what? The more micelles you have, the faster you shrink. The faster you shrink, the faster you go. So you disappear faster, but let's see what happens to the trajectory. So this is low, and this is high concentration. You can't really see anything by eye, I would argue. But if you look in the experiment, the devil is in the detail, often in physics. And so what you learn, this was your question before, right? What you learn is that the noise is roughly 1,000,000 times hotter than the room temperature. It's a hydrodynamic noise, but it's really, really hot. And so you actually, even on pretty pretty slow time scales, you see the diffusive regime. So you see it be diffusive, ballistic, and then maybe diffusive or log T up here. And so what's also nice is that the theory completely predicts this trend of what happens to the mean square displacement, and the only thing the theory knows about it is, the only way it enters into the theory is the shrinkage rate. So that's kind of nice. It shows you that all the data can be fit with this model. So what happens to the effective temperature? The effective temperature, if you don't know anything about the mechanism, looks like, this is, if you just say that you think that the noise can be related to temperature in the classic way, you say, oh, I go from 0.1 times a million, 100,000 times hotter than the room temperature, not quite a million, down to 1. But then what you realize is that at higher Peclet number, you have higher hydrodynamic modes. This is like a whole other story. They get excited. So you could argue that the switching becomes more and more fast. What's that? Oh, okay. A shooting or
Fireworks.
excellent. That's good then. It's a celebration of droplet swimmers. Okay. But then we thought about it for a bit longer, and we discovered that, in fact,
It's with our invited speakers, when they tell that things are working, we
there
just
is a
celebrate.
fireworks.
Yeah, you
There's
see.
fireworks. Excellent, excellent. We're celebrating the success of the model. Great. So remember how I was excited about this volume term? It's because what we thought was decrease in the temperature or something can really be explained by a stupid thing, by a simple thing. And that's the idea that the higher the concentration of SDS, the larger the micelles get, and this has been measured by NMR, and no NMR, yeah, NMR. I was going to say MRI, no, NMR. And if you look at this data, which is perfectly in agreement with what is published, you actually get that the coupling strength remains constant over the entire range of SDS. And the only reason why we see a number that's decaying is just because we didn't take into account that this delta is getting a little bit bigger when the SDS is gone. And so this is the Houdini moment. I don't know if you like magic, but one artifact of this swimmers, who was talking about 3D? You were talking about 3D. That's why I was like, I won't say anything more at that point. One thing that's nice is that there are mirror charges. So because these droplets are next to a surface, the fact that there is self-avoidance means that there is a repulsive force away from your own trail, but it also means that there is a repulsive force from the surface. And so what you end up doing is you end up levitating. So if you now don't take a swimming droplet, but you go to the regime where you, remember below the CMC, you could be in a regime where you're shrinking, you're creating a gradient, but you're not moving because you don't have enough oomph to move. What you see is a droplet that will just spontaneously rise and rise and rise until gravitational potential balances out the repulsion force. So you're being repelled from the surface, and so it's a nice way to actually have systems that are unaffected by the surface. You can just use this. It's also a nice way to use gravity to measure this force and show that the agreement with the model. It's another way to show it agrees with the model. So here is the Z, the levitating height of a droplet. This is what a reference dead droplet would do, and this is what the hovering droplet would do because of the gradient. We actually measured the mirror in the confocal. We made a little droplet experiment in which we can measure the height, so that was a really fun experiment to do. And I'm going to end by saying that you're back, even though we've understood the lifetime of one droplet, which dies when it runs out of fuel. When does it run out of fuel? When it's completely dissolved, so it literally disappears. You're in trouble again when you get to the many-body system, because as soon as you put 2 droplets in here, not only do they repel their own trail, they also repel everybody else. So if you inject 10 droplets simultaneously at the center, they hate each other. And so you see this hatred by the fact that they all go to the end of the Petri dish, and then you see them do their thing here and so on. And so you can watch, you know, how they do that. But you're in trouble with how to analyze these guys. Because a single droplet, what I spend time explaining is this guy, and 10 droplets is now super diffusive because they hate each other, and 25 is even more. And so, you know, we don't know what to do with a many-body system of these swimming droplets, but at least it's a start, you know, at least it's a beginning of understanding. We don't understand really the hydrodynamic noise deeply, but people are working on visualizing the flow fields and looking at these, which modes are excited at which Peclet number and so on. And yeah, we claim that this coupling strength and effective temperature is measured for the first time. This is a slide for Carolina, which is, I want to watch The Meaning of Life with her because she still hasn't seen it. She told me that the other day. And I feel like that's been my course, because I feel like the movie is, I don't know how many of you have seen it, but the movie is split up into 7 parts, and the first part is the miracle of birth, so that was giving birth to Foldamas. Then growth and learning was who likes who and how much between the droplets, right? Then there was fighting each other, the origami speciation with the giraffe, right? And then death is the droplets dissolve. So I feel like in these 3 lectures, we basically cover all of life, no? Life, death, and beyond, you know. Thank you very much for listening.
I don't need to watch the movie then. You
Huh?
know, you already explained me the meaning of life. So I
It's
know I
true.
don't understand movies, as I told you.
It's true.
In particular,
there's a few
month-
things missing, like the autumn years and organ transplants and things. We didn't talk about that, but
So any question? Okay, there first.
yeah, sorry.
Thank you very much for this beautiful talk. My question is, my first question is if this droplets is, I don't know, for me, this system is very similar to Janus particles in the sense we emerge in self-propulsion direction doing a specific mechanism which generate a kind of polarization inside the particle. In Janus particle, when we increase, for example, the density, as the particle avoid each other, as the system, we obtain a liquid to crystallization transition. Did you check if you increase the density your system, we can obtain some similar behavior and liquid to crystallization?
So actually, it's a whole other story. But what you'd predict from the model is not that, because you have, you have repulsion in the trails and so forth. So you might imagine that they just collide and move away from each other, and that's that. But actually, counterintuitively, what happens is, actually, Anton, you had a slide on that. You get capillary forces because of hydrodynamics, because you're squeezing liquid out from in between the droplets, which makes them stick together. So in fact, when you put many of these guys together, they do have a short range attraction and a longer range repulsion. And there is an interplay between those two that does create actually a pretty fascinating result. crystallites, clusters of a very specific size. And if that size is 16 droplets, if a 17th one joins, one is expelled. It's a very funny system.
Okay, before you go to the other question, where is the list of attendance? Okay, can you pass here, please?
Thank you so much. I was wondering, I don't know if I missed it, but you talk a lot about the water and the soap and the oil. I was wondering if you have already tested with other combinations of like media, like that had the same mechanism, but maybe instead of water, like with a higher viscosity, and if this viscosity could like suppress the superdiffusion that you mentioned or something.
Yes, so that's a really good question. There's lots of chemistries, including like, which are not DEP, which have actually, there's a woman, Lauren Zaza, beautiful paper just came out about predator-prey droplets. So now you have this story of the repulsion. You have a different chemistry in which now the micelles like their own excretion. So they go towards their own trails. And so now you can make really cool systems in which, you know, everyone's buzzword is non-reciprocal reactions. So you can get really fun physics out of different chemistries of different emulsion species. Good luck to those who want to study that. That's what I will say, because this system with 3 ingredients is a mess.
Okay, thank you very much. It was very nice, and I learned a lot. And my question goes to know if, while putting the drops, those are oil droplets, was the domain restrained? I imagine a situation where I put 16 drops, for example, in a very large domain that has the same characteristic. That's water with oil, and yeah, the specifications you gave. But I expand it. Imagine the size. Let me just imagine something very big in this class. What will happen? Will the
Are you
drop?
saying does the swimming pool size matter?
Yes.
No, not on the scale that is larger than about 100 droplets wide.
Okay.
Are you asking if there's an interaction with the surface or the border?
No. I'm asking whether the patterns that were formed due to repulsion between the oil droplets,
Yes.
are they still going to be like, because I saw they went to a certain level, then they started coiling. I'm
Yeah.
asking if maybe we expand the arena, maybe they will just go straight to, I don't know.
Oh, I see what you're saying. So
Okay.
there could be, in the very last movie I showed you, right, there was a finite size effect of the swimming pool where what happens in the mean square displacement is that you end up with a plateau because they've got nowhere to go. And you're right, we should expand the swimming pool and show that it goes further. But I was thinking about something else, but you're right.
Thank you for this nice talk. I have a question about when you feed this mean square displacement, at the end of your data you have like undulations. Why you have these undulations?
Undulations?
Or like, it's not regular, the data, I think.
Oh, I think it's statistics. I think when we get to long times, we don't have enough statistics. Do you mean this guy?
Yes.
This? Yeah, when we get to like 100 seconds, since they're only 200 seconds long, and since we have ensemble average data instead of time average data, we start to get into the noise there.
Okay.
But that's kind of all it is. And then there's finite size effects too, a little bit. That's why this is a bit flatter than diffusive.
Hi. I had sort of 2 questions, comments, which is sort of the trajectories here and this fact that you get less and less self-avoiding, I guess, with time, kind of reminds me of a blob picture of polymers, right? Where you have sort of self-avoidance on different scales. Do you know if there's any
Yeah,
connections
so
there?
actually I didn't show you the slides, but I'm not sure I want to. So the ABP model and like the, what's the other one? Run and tumble. They have equivalents in the polymer literature for worm-like chain and freely jointed chain, based on what assumption you use for the excluded volume, things like this. For when you get to the our model, you can think about this as something called the true self-avoiding random walk, which is, I don't understand it quite, because it means that there is a finite probability with which you can cross your trail. So being true means that you can go back on yourself. Once you make that and you look at infinite time, you go back to being diffusive, or at least going with the log of time. And so it becomes really hard to distinguish between diffusion and this correction. But for those systems, there are analogs in the polymer literature too. Shura was telling me some Russian name for the model of the self-avoiding polymer that can reptate on some time scale, and I forget. Could it be the Kratky-Porod model or something? I don't know. But you're right. It's a literature that is heavily based on the polymer analogies.
Thanks. And then my other kind of comment along the same lines is there any connections with Faraday droplets? You know, they're kind of, you could make droplets bounce on top of a vibrating water surface.
Yeah.
And, you know, there you also have the droplet dynamics kind of coupled to a medium. And I guess the medium dynamics is different. But
I think it's very complementary. I don't think the details are the same, but I think these Faraday, the bouncy droplets or whatever, they're also based on this relationship with the drive. And so maybe there is a model that could be made that's somehow analogous to this model.
Thank you.
I don't know.
Any other question?
Yeah, here.
May I ask about the first part of your lecture regarding
sure.
the replication
Yeah.
system? You mentioned that making a replica of a long chain doesn't work in the lab yet.
Yeah,
I guess
exactly.
you are working on that. Can you comment what are the
it's actually
theory that you have behind the not
the
working?
theory is not a problem here.
I mean, what
The
you
theory-
speculate is the reason for not working
So it's,
yet.
I think somebody here mentioned, it's the fact that you can have non-specific interactions. And so you really want different energy scales for the horizontal and the vertical. You want to have different species of DNA that connect the sequence of the mother to the daughter, right? And you want this idea that temperature and light don't affect the same bonds. So it's just about designing. I don't think there's any a priori problem with doing it. It's more like time, number of people in the lab. Like, I think it's more like we need to put an effort to make that work, because all the attempts were done with solid colloids. And as I mentioned a million times, solids get kinetically trapped, and they can't freely rotate, and they can't slide past each other. So you end up with fractal aggregates that some of you love. I get that. We don't. And so we haven't tried this seriously with droplets. And my hope and my conviction is that it will work, because droplets seem to know how to rearrange.
Hi. I would like to understand better the parallel between this model of the droplets and the cell. It was one of the first slides,
Yeah.
if I remember.
I mean, the cell I didn't even, I was hoping no one would ask, but we know that cells divide. We know that the genome gets replicated. We know that there is a complex machinery that does that very well, right? And so the idea is simply to say, if we borrow from biology to do, for example, this polymerase chain reaction, then we know how to do self-replication, but we're borrowing the machinery that evolution has come up with to do that. And so the statement was, is there a way that we can start with inanimate objects like colloids or droplets and don't use enzymes and don't use active matter and just use daylight and temperature fluctuations that are natural and end up with exponential growth of this colloidal material. That's never been done still. And like I say, I think the obstacles are more technological than fundamental. But that's the only analogy I try to make with the cell.
You mentioned in the second part, I will make a rude comparison. Like now
Yes.
I will switch from cells to droplets.
Yes.
So this connection, I didn't
Oh, there I was
understood.
saying I was going to be rude by saying that a droplet is a cell because, of course, when a neutrophile goes to eat a bacterium, there's a lot going on, right? Like the cell needs to eat. It's a much more complex system. So by making
okay.
a claim that a solute-mediated interaction that follows the inverse square law between two droplets may have, in fact, nothing to do with the gradient interactions between. That's why I said it's rude, because it might have nothing to do with what happens in biology.
Okay, thank you.
But it's a beginning, let's say.
Okay, if there is no more question, I'd like to thank again Jasna for those three
Thank you.
very nice
Thanks a lot.
lectures.